Okay, lastly, we make a tentative conclusion about what we think is going on back in the population based on our sample. And I say tentative because we know that there's sample variability, so this is going to cause issue because we know if we were to do another sample of this data, we could get quite different results. And so we have a bit of a dilemma. But based on our sample, we can draw a little bit of a tentative conclusion. So we say, based on this sample, based on my sample, it appears that there is no difference between the male and female median systolic blood pressures. And that sort of relates to your question. So you basically answer your question that you posed earlier. The sample shows lots of overlap and no median outside the box or the other. We talked about making the call, and you may have to check your notes on what that means, making the call, about whether you can decide whether there's a difference, and that has to do with the position of the medians inside each other's boxes. So the fact that the median falls inside this box and this median falls inside this box means there's lots and lots of overlap, so that there's not much difference between these. So we can't make a call that back in the population that there's actually a difference. This suggests that back in the population there's no difference in the median blood pressures of males and females within this British study. The problem is that that's not where we stop our investigation. This could change. This idea could change because we know that our sample could be very different from somebody else's sample. So then we go on to talk about sample variability. What is it? How does it affect our conclusion? And so we talk about those things that we have spent a fair number of days talking about. However, due to sample variability, I know if I was to take another sample, it would be different. And you should say how it would be different. It would be different in the interquartile ranges. The medians and means could be quite different from the sample. Sample variability can't be eliminated. It can be reduced by taking a big size sample. This sample was 200, so it's a good size. And because of sample variability, further investigation is needed to confirm our findings. And this is where we start getting into the second half of our internal, where we talk about our confidence interval, and we start formulating using Enzyte a formal confidence interval using a method called bootstrapping.